DNA mutations, skin cell death, and possible link to cancer. According to a new study, using a UV nail polish dryer might be more dangerous than we thought. Unlike regular nail polish that begins to chip and fade within a matter of days, gel nails can weather the wear and tear of everyday use for weeks. In many ways, gel nails are like a 3-layer cake. Made up of a base coat, nail polish, and a top coat, our nails are set to bake between each layer under a UV light for a couple of minutes in order to dry. While a UV nail polish dryer is a key component in the gel manicure process, there hasn't been much research into the effects it may or may not have on human health. The co-author of a 2023 study published in Nature Communications looking at the effects of UV nail polish dryers on skin cells told UC San Diego Today, "If you look at the way these devices are presented, they are marketed as safe with nothing to be concerned about. But to the best of our knowledge, no one has actually studied these devices and how they affect human cells at the molecular and cellular levels until now." The inspiration for the study came after Alexandrov learned of a young beauty pageant contestant with a rare type of skin cancer which had developed on her finger. The study team used 2 types of human skin cells as well as a sample of mouse cells and subjected them to 2 different UV light exposure scenarios. In one scenario, the cells spent 20 minutes inside the UV light device and were then removed for an hour before undergoing a second 20-minute round of exposure. In the second scenario, cells only underwent one 20-minute round of UV light exposure, but did so once a day for a total of 3 days. Regardless of whether the cells underwent acute exposure or chronic UV light exposure, cell damage, cell death, and DNA mutations were observed in both instances. More specifically, the observed DNA mutations resembled those found in patients with skin cancer. The research team noted that these findings suggest a possible link between the use of UV nail polish dryers and a greater risk for early onset skin cancer, particularly in the hands.
There was a potential link between these machines and cancer.
In a press release, one of the study's authors stated that she has since opted out of any further use of gel nails upon learning that DNA mutations and cell death took place in the immediate aftermath of just one 20-minute UV light session. Furthermore, findings also showed that 3 consecutive 20-minute sessions in the nail polish dryer resulted in up to 70% cell death. However, the researchers emphasized that the study only illustrates correlation, not causation. Far larger longitudinal studies would need to be completed before a definitive relationship could be confirmed to the public. In spite of this convincing research, some experts suggest that there's no need to swear off gel nails entirely. Instead, fans of the method are urged to consider implementing some protective measures during their next manicure appointment. For instance, dermatologist Dr. Shari Lipner told NPR that gel nail fans can add significant protection by utilizing sunscreen and fingerless gloves during their sessions. Scaling back on how frequently you visit the salon throughout the year could also have an impact.
Radiation of UVA causes damage to the skin cells.
When it comes to safer alternatives for drying gel nails, you might think that LED lamps would better protect you from potential skin damage. Although there are LED lamps out there that do not give off UV radiation, they wouldn't get the job done. This is because gel nails require UVA exposure in order to dry or cure, making it difficult to avoid when you're getting a manicure. Furthermore, although our nails require less time underneath an LED lamp compared to a UV nail polish dryer, that doesn't necessarily mean that these devices are safer. Rather, the UVA ray exposure we get from an LED lamp is more powerful than that emitted from a standard UV lamp. This exposure is especially problematic, as Today noted. UVA rays are the most mutagenic wavelength range of the UV spectrum, penetrating the skin more deeply than UVB rays and playing a role in skin cancer development and premature skin aging, such as wrinkles and sunspots.